Let's talk phylum Porifera. These are the sponges. And porifera means pore bearing. Yeah. Sponges are defined by their pores because sponges are water filtering organisms. And we're talking about living sponges here. The bath sponges, at least the natural ones, are basically the defleshified skeletons of sponges. So you're just taking the dead body of something and rubbing it all over your face because it feels nice. You complete sicko. Yeah. What a weird thing to do. Stop it. Here are porifera, and so the the pores that we're interested in are ostia, ostium singular, and these are where the current comes in. So it's going to come in on the sides of a sponge, and then there's going to be an osculum in the center, and that's where it's going to go out. So the osculum is like a big chimney, and shown up here at the top, our little mascot for today is they're putting fluorescent dye, fluorescein, in there, and you can watch the sponges slurping this up and then slurping it in from the sides and spitting it out at the tops, because that's how sponges work. This is how you can tell if a sponge is alive. If it's doing filtration like this, it's working pretty hard at that, and yeah, honestly, it makes a lot of sense. Like we said in class, blue whales, all those big baleen whales, I don't know what they're thinking. Like swimming around, doing all that work to catch up to the krill when you could just sit there, rooted to the bottom, spin your little cilia around, pump water through, and have all you can eat all the time without even getting up. Isn't that, in some way, the American dream? I don't know. Maybe it's not your American dream. That's okay. Country can accommodate more than one dream, I'm sure. So let's take a look at these guys. They're pretty neat. The important cells in sponges, there's going to be epidermal cells that are going to make a sort of a coating around the outside. There we go. They're going to form our nice little spongy exterior. And their job is to keep parasites and bad things out, and also to create little lanes where the water can be pulled in, because we don't just want to move water, we want to move water past our feeding apparatus so that we can, pick out all the tasty bits. And so here are they're called collar cells, or choanocytes, which I think is just Greek for collar cell. And they have a little collar of sort of sticky tentacles. Those are going to catch the food. And then they have a long sort of like a flagellum, eukaryotic one. And that's going to spin around, and that is going to generate the water current. And all of those are going to be synced up. So they're all going to be spinning in time in roughly the same direction. And they're going to be pulling the water in one particular direction, in through the sides. And then it's just going to crash around there and get filtered as much as they can. And then it's going to go out from the center. There you go. Just like in the picture. Other interesting cells, they're going to be cells that make these little sort of spiky bones. They're hard parts. Sponges have some of the most primitive skeletons that we know of in the fossil record. And we think the record of sponges goes back to the Neoproterozoic, so think everywhere between 1 billion years old to about 540 million years old. So in that sort of zone, and people argue about which one's the oldest sponge and how spongy it is and whether that even has any meaning. Yeah. So those are the cells, and so there are specialized cells that form the pores. There are specialized cells that sit in between the epidermis and the collar cells. These amoeboid cells are called archaeocytes, and they are going to be something like a sponge version of a stem cell. They can turn into any of the other cell types, and they're going to populate the middle, like the bulk of the sponge. All right. Here's the oldest bona fide sponge fossil, as far as, that most everybody can agree on. There are older sponge-like fossils that go back to something like 800 million years ago, so 200 million years before this. And we will look at those later because they are interesting. They may not exactly be sponges, but the term sponge probably has no meaning back at this point because there's nothing else. This is just eukaryote that filters water like all the other eukaryotes that filter water at the time. This is before, any of the modern animal phyla existed. And so, what is this thing? Well, you can see it's got a big hole in the center where water could go out. That's cool. That's sponge-like. It's got a bunch of little tiny gaps in here, which maybe water goes in there, or maybe these were just little sort of domed structures that have been broken during fossilization and preparation. Hard to say. Those are both possibilities, but you could see little gaps between them, and those could be where water could have come in. So if this is a sponge, this is how it would work. That's kind of what this paper is saying. On the inside, though, there's this really fascinating texture. It's like a little honeycomb. And there's a ridge that runs on the top, and there's like a little ring. You can see here is a little ring that goes in there. And so there are, they're imagining that these are the little seats. It's like when you look at a stadium and nobody's there, you can just see all the empty seats where these little very soft-bodied cells would have been. And these would have all been hard parts, or at least kind of leathery parts, a little more durable, more likely to fossilize. And so, here we have our little color cells and our little cilia, I guess is a better word for those. They would be generating water flow. So this may be correct, looks reasonable, but at the same time, it is open to other interpretations, as is pretty much everything in paleontology. I think this looks like a reasonable case for the first confidently spongy fossil. Now, these are coenocytes in a sponge, and you can see in here, this is one of the little chambers that water is going to be drawn into. And look at that chaos in there. So here are the colors. These are what's going to be capturing the little particles. And here are the flagella whipping around, all kind of tangled up there together, because this is a dead one they photographed. But those would be all sort of functioning, whipping in time, and pulling water through. And so it's going to pull water maybe from the outside is here to the inside. Can't really tell what's up or down in this, but those are the little collar cells. Now there exists in biology a single-celled version that looks a whole lot like the collar cells in a sponge, and it's called a choanoflagellate. And here's a picture of one. It's just like a little champagne glass with a little tiny whip that it's thrown around at the top. They exist by generating a little water current and catching particles that come in as a result of that water current. So they're drawing water toward themselves based on the shape and motion of their little flagellum here. And they can live alone. They do start out alone, but some kinds will actually live in little clusters, little tiny clusters. So they're mostly single-celled, but a few of them have like the start of multicellular, I don't know if you want to call it technology or lifestyle. Those both sound weird when applied to non-human things, I think. But essentially, sponges are only loosely multicellular because the cells themselves can reconstitute an individual and differentiate pretty well, and they're not necessarily dependent. Like a sponge doesn't have an arm where if you cut off the arm, it's dead, it bleeds out. They just have more sponge, and all the sponge has a lot of the same stuff, just different chambers with more or less the same thing going on in there, which is whipping and catching the particles. Fortunately, we have the genome of, I think it's Monocyga brevicolis, I think is the name of this guy. So that's the MBRE here. And that is the a choanoflagellate. So this is the single-celled, we think, primitive relative. These would be more primitive than comb jellies, ctenophores, more primitive than sponges, more primitive than placozoa. So these would be like the last common ancestor they would share with all the metazoa, the metazoa being over here and choanoflagellates being here. It's interesting that they look just like, not ctenophores, not placozoa, but porifera. So you've got an ancestor, and the ancestral traits are retained in Porifera, but they've been lost in these other two branches, which we think actually come in between. And so that's always neat when that happens. You think, wow, you must have made a really big change in evolutionary terms to get here. And so here they're showing the different animals. The H sap is Homo sapiens, and that's up here. We put ourselves first because that's what it means to be human. Yeah, we're number one. Thanks. And so Nematostella vectensis is the Cnidarian. And so here's Drosophila melanogaster, the little itty bitty fruit fly that everybody loves. And Cyona intestinalis, I think if I'm saying that right, is a little, it's a star ascidian. It has a little spinal cord, but in a squishy body, and it doesn't have any bones. So it's a chordate, but not a vertebrate animal, if that makes any sense. Okay, so they're shown here with little boxes and colors what we found and what we didn't find. So dark is found and white is didn't find. What they do find are a bunch of these things. And down here, you've got fungi. So these are all these little brown guys. And you've got a slime mold, Dictyostelium, and you've got a plant. And so these are things that would be outside the animal kingdom. Yeah, kingdom is one of the stupidest words in biology, but it's there, so we'll use it. It's just that it doesn't mean what people think it means. And so, fungi generally lack these, but ancestors of all animals, like Monocyga, or things related to the ancestor of all animals, and all animals tend to have them. So these are all proteins for cell adhesion. These are things you would need in order to be multicellular. And we find the proteins in these pretty much single cellular, effectively precursors. They're from their modern times, but they have retained their old structure. And the idea is that this probably reflects the genetics of this organism right here, where the branch between all the normal animals and the conoflagellates makes. So the last common ancestor of those things would be right here on the tree. Great-great-great-great-grandparent level by the look of it. So they probably had proteins which were later repurposed for cell adhesion. So in a single cell, you can imagine those proteins being on the outside, and they can provide some sort of coating. They can stick you to the environment, for example. They're rooting this thing down, at least in part. But yeah, you can see later on, all you would have to do is express those same proteins, but have two cells there, and then those proteins may interact with each other, or they may interact with something and mutually bind to the same thing, and you can have organization, multicellularity developing. In here, these are extracellular matrix proteins. So these are things you would normally find on the outside of an animal cell. They tend to be rare in fungi. There are a couple things that are conserved in eukaryotes in general. But they tend to be pretty common in animals, and we've got almost a full set of these in little Monocyga right over here. And lastly, down at the bottom, these are developmental genes. So Hox genes are the things that determine how many fingers you have and where they grow, for example. And like big stuff, like whether your head ends up here and whether you develop a brain inside of that. Yeah, all the things involved in developmental patterning are going to be genes like this, like Hox genes, for example. And so you can see there are some genes that we do not find in Monocyga, but a surprisingly large number of these genes. Forkhead is something that is used in animals, but including flies. I believe when you mess this up, you get a fly with a split head like that. Homeodomain genes are used in developmental patterning and would be similar to Hox genes. These are homeodomains of the X chromosome, I think. I don't know. You'll have to look that up to make sure. We have a range of things that will be reused later to pattern different body parts. Right now, what are they patterning? Probably nothing. Yeah, they are just turning on different groups of genes, which is really all patterning groups do. And so these are probably controlling different environmental response factors like, oh, it's cold outside. Let's put on a coat of more extracellular matrix proteins to stay warm. Something kind of dumb and basic biology like that. And so, yeah, you get the same things, and these are going to be reused to say, instead of just like getting warmer, let's grow an arm over here, and then we can, reach out and move around and grab stuff, and yeah, put that Big Mac in our mouth where it belongs. Yeah, I'm getting hungry. You can tell this is lunchtime. All right. So here are some of the proteins in Monocyga. And tubulin makes microtubules, so okay, we can see that's making up the little flagellum here, so that looks good. Synaptobrevin and SNAP25, and I believe MUNC18, I'm not as sure about that one. These are proteins that you would expect to find in the synapse of a neuron, where one neuron connects to another, and they transmit an electrical signal from one cell to another cell. This is essential in thought and in nerves. And some of the proteins are conserved all the way down to the single-celled choanoflagellate. And they're showing you in here where these are. The synaptobrevin lines up in a kind of a cluster here underneath the cone and around the edge of the cell. The SNAP25s are definitely located up at the flagella side, but they are not on the membrane. They're just sort of clustered up here, just like they are in a neuron. If you were to look at that, that's where you would find the equivalent of SNAP25. Yeah, and the MUNC18 is roughly in the same place. So they have the things that will eventually be reused for nerves and brains, even though it's still a single-celled organism, because that's what evolution is. It's reusing components you already have. Often it's taking something that had many different possibilities and losing some of those until you just have, one collection of those possibilities that's still pretty well adapted to some environment. And then, all of a sudden it's all about getting food resources, finding food and shelter, just like now. Yeah. Okay, let's talk sponges specifically. The biggest and most important group of sponges, because they are most of the sponges. So I don't know, important's a weird word. We won't say that. Just the most numerous group of sponges are Demosponges. Here are some examples, and Demosponges kind of run the gamut. They have, these are the spicules from different Demosponges, and look at the variety here. Just all kinds of stuff. They tend to have 1, 2, or 4 axes. So this would be like a 1 axis, a 2 would be like this. They're going this way, then this way. And 4 or some multiple of 4. This one I think is an 8 right here. So it's 2 4s back to back. Yeah, some of these are balls, some of these are crazy shapes, and these are going to be specific to a particular type of sponge. Each sponge will have potentially a range of spicules that it can make, but those spicules are going to be characteristic, and these are spicules of Demosponges. Now, these spicules in Demosponges can be made of the full range of things that sponges use. Some of them use a protein called spongin, with the ending of protein next to the word sponge. Just sort of smush that together, and that's a flexibendy protein. When we make bath sponges, we boil away all the flesh, all the porocytes and archaeocytes from the sponge, and just leave the nice skeleton. And we do this with sponges that are mostly made of spongin, and that's the nice thing that feels good when you rub it on your body. Yeah, you weirdo. There are kinds of sponges, including Demosponges, that can actually make silicon dioxide spicules. And so those are literally the same as glass. So, yeah, kind of interesting. And they are mostly found in shallow marine environments, but when you see these deep ocean explorations, a lot of what is down in the deep ocean, there's far fewer things down there. There's far less nutrition going around. But you do tend to see that those deeper environments are quite often dominated by sponges. It'll be a mix of Demosponges. There'll be rarer and Hexactinellid sponges, which we'll get to in a minute. Those are going to be common. Yeah, and they were important Paleozoic reef builders. Before there were coral reefs, there were sponge reefs. Yeah, imagine that. That's weird, huh? Yeah, but history is weird. These are just some carnivorous sponges, because why not? So these do not pump water and capture particles for food because they live in the super deep ocean. They actually have something like a root system that will reach down into the mud. And so these things, if they'll just sort of release lots of little babies, and I believe these are believed to be breeding structures here, just releasing babies all the time. If the babies happen to fall on an area where something has died and rotted in the mud, then they will be able to grow there, extract nutrients, potentially for thousands of years, and just broadcast, send out more little babies. Most of them are going to fall on stony ground, and they are not going to do so well. They're going to die. But every once in a while, and if you're there for thousands of years, it's probably going to happen. Yeah, then they're going to end up starting the next generation. So yeah, these are Demosponges, these particular ones. And these are found exclusively, as far as we know, in very deep water. Yeah, kind of low nutrient, low competition environments. All right, so let's look at sponge genetics. Why not, right? So shown here, they've got a bunch of different genes. So like everything in this section, you got your ion channels, your voltage gated ion channels, your G protein coupled receptors. These are things that would be used in thinking, in nerve function, for example. You got hormone receptors, gap junctions like you would have in the heart to pass electricity along. You've got things that would be used in the immune system, like little hormones and cytokines. That's like communication between white blood cells. Sponges don't have white blood cells. Sponges don't have, as far as we know, anything that's very similar to an immune system. They'll have functions that will probably end up doing some of the same jobs, but maybe in a less sophisticated way or less complicated way. That's probably a better way to say it than we would find in people, for example. The colors here are going to be counts of the number of genes, the number of copies of this type of gene that you get, because that's the other thing that happens in evolution. You lose some genes, and other genes, you make more copies of them. And now that you have multiple copies, maybe one can keep you alive, and maybe the others can mutate ever so slightly with no real penalty, and they can end up binding a slightly different molecule, doing a slightly different thing. So gradually, you build up the ability to bind more things, to, do more kinds of chemistry inside the cell. And that's the other way that evolution works. So here's HSA, that's people down here. AQU is the, Amphimedon? Queenslandica? Something like that. Anyway, that's the name of the sponge. MBR is our little monocyga. This is our coenoflagellate. And so this is our little tree of life. And here's Arabidopsis. That's the plant. And there's Dictyostelium, which is the little slime mold. And so these are eukaryotes, but yeah, not our kind of eukaryotes. And then everything from this branch onward, I would say, is an animal. And then we get into metazoa once we're from about here onward. So yeah, over time we get duplication of some of these genes, in particular in Homo sapiens. So we don't have more of anything. We have more copies of some of the same things. And that's what makes us special. Yeah, genetic plagiarism, I guess. I don't know. We're plagiarizing within our own genome, so I guess it's okay. We'll allow it, yeah. Here, this is just showing what you get and the origins of, like, the first time in the genetic record that each of these things appears. Because they all appear, these are the proteins that are involved in a synapse. So here's the synapse that's sending the signal with our synaptic vesicle. And things like SNAPs, there's our SNAP25. These are synaptic vesicle tethering and tethering proteins that will, move them to the right place and make sure that they get released at the right time with some help from all these other things. Down here, we have all the receivers of those neurotransmitters, which are going to then generate a new action potential in this cell and send the signal on down the line. And all this is going to happen so fast that it's, the speed at which you can think. Like if it was really slow and somebody threw a ball at you, there wouldn't be time to say heads up, and then you look up and say, there's a ball there. Maybe I ought to put my hands up and catch it, and then, you catch it. No, we'd be walking around with far fewer teeth. Yeah. Based on all the balls that were flying around, at least in my childhood. Everything that's blue is present all the way down to the bottom of animals. So think down to coniflagellates, for example. Below that kind of animals, you have things like pisticonts, which are a particular kind of animal-like organism, but would include, I believe, the slime molds, although we'd have to go back and look to be sure. True metazoa are going to be things like sponges and comb jellies on up. And so these things appear for the first time. But you'll notice that there was already an ancient eukaryotic version of a neurotransmitter receptor there. We've just made more copies of it or slightly different versions of it. But these are proteins that are probably related to each other and do the same job. Yeah. Second type of sponges of great consequence, Calcarea. These have a calcite, that's calcium carbonate, skeleton. Sometimes it's a massive calcium carbonate. It's just like all fused together, great big chunks of it. And these tend to be really heavy sponges, really thick, but they're going to have little channels that run through them for water to go down because they're still sponges. They still live by filtering. They're just going to outsource the work that was done by their epithelial cells and say, you know what? A piece of rock could, form a barrier and would do just as good a job. But yeah, you're out of work. We're outsourcing this. Their spicules tend to have 1, 3, or 4-fold symmetry, but 3-fold symmetry is the most common. Like here you can see all these little tiny Mercedes-Benz logos. Most of them upside down. Yeah, why is that? And they're going to make up the sponge. And these were super important Paleozoic reef builders. We think the earliest sponge fossils in the Cambrian, anyway, had calcite skeletons, but they did not have spicules. So they did not have these little spiky individual bones. They just had big, thick walls of calcite. So some of these Calcarea that retain massive skeletons, they'll have little spicules inside, like as a sort of a genetic throwback or a developmental thing. Like first you grow the little one, then you sort of cement them all together to make the whole. Yeah. But yeah, they were big time reef builders. And because they're already made of beautiful calcite, big, chunky, durable, and rooted to the bottom, if something comes in, sort of a mudslide, covers over the bottom, then you will see that those things are going to get fossilized very nicely. And so there are places where we get a lot of sponge fossils. Yeah. Including some of the places we're going to go in the course. All right, there are other groups of sponges. These are the homoscleromorph sponges. These are similar in most ways to Demosponges. Like if you're just looking at them, if you look at their genetics, though, they're considered to be a separate class of sponges. It's a cryptic class, though, in the sense that by looking at them, you would say that's probably a Demosponge. And you'd be right, it probably is, but if you look at the DNA, then you find out you're wrong, and it's not quite. So some fossils that we are identifying as demosponges may be homoscleromorph sponges. We have no idea how these things evolved because you can't tell them by appearance from other sponges. Now, they may have molecular markers, and with what we're learning about molecular fossils, we may actually be able to figure out the answer someday. So, ooh, maybe. Exciting. Here are the neat sponges. Hexactinellid. Hexa, like 6, hexapod, whatever. Actin means like little spines. So they've got 6 little spines coming out everywhere they have a spine. So here's one central one and 6 little ones that go around the edge. Here's a central core and 6 little identical branches coming off of it. These are hexactinellid spicules. Oh, and there's also 6 little clusters of 6 coming off of there, because why not? Yeah. These are examples of these glass sponges. You find them particularly common in deep ocean water. Interesting thing about these is that they actually don't have very many individual cells. They have a lot of cells, particularly in the body of the sponge, that are fused together like you would get in a slime mold. And so there's a bunch of little nuclei, thousands to millions of nuclei, and it's just one giant cell, and they flow around. And the nuclei will, take turns expressing different proteins, and different ends of the cell might be making different products, but that's okay. Who says cells need to be separate? Why can't they just be one giant blob together? They'll have some cells that are individual. I think a lot of the collar cells on these tend to be individual cells from their evolutionary origins of basically being single unicellular. Yeah, but these do exist in the fossil record. They tend to be more delicate, at least the modern ones, and so they are generally rarer than these big chunk sponges, which are going to be like the Calcarea or some of the Demospongea that are basically built like a Calcarian, just, tough, thick little things. Where do you find sponges? On the coast, you find a lot of sponges. This is color-coded by diversity. But you see these little spots out here in the middle of the oceans. You do find a whole lot of sponges when people go down and look. And there are probably a lot more sponges there. You see a few spots where there's a 0. So somebody actually looked. They didn't find any sponges there. Hey, happens sometimes. But there's probably a lot more sponges out there. They are not necessarily in very accessible places. Like, look at the numbers. Hundreds of different species of sponges surrounding Antarctica. When's the last time you went to Antarctica? And when you went there, did you spend a lot of time under the ice? Yeah. In the ocean? Were you counting sponges while you were down there? Yeah. That's basically why we don't know quite as many sponge species. They don't do quite as well in modern tide pools. Although, yeah, if you were to go to John Pennekamp Coral, whatever it is, State Park, National Park, whatever it is, which is in Florida, the corals are effectively extinct in the wild in that park. It was set up to preserve them, and the climate changed and pollution and everything else, and a couple of bad years, and bam, no more corals left in the park. You got to go quite a ways outside of the park to get corals you can see. Except in the gift shop, where they have a couple of corals living in tanks. Those are the corals of the coral park. But what you get there are tons of sponges moving in. And there's some algae, but the sponges seem to be doing well there. You'll see these giant orange barrel-shaped sponges and sort of pinky ones. And yeah, they're doing fine. They fill a similar ecological niche to corals. And sponges and corals can live together. But when there are conditions that kill off the corals, it's still a prime spot. It's still close to the surface. There's still plenty of phytoplankton, and so stuff moves in. Yeah. Is it less spectacular? I don't know. I like sponges a lot. I'm pretty fond of corals, though. So, yeah, maybe it is. Yeah. Okay. Here's a little summary thing. I think I pulled it off of Wikipedia. It's just like, what do you find? Most kinds of sponges, you find single cells, hexactinellids, which are our glass sponges, because they make glass spicules with 6-fold symmetry. Those tend to have multinucleate cells, and so our word for that is syncytia. And this is also what you get when cells get fused, like when you get cells that are infected with a virus, and the proteins on the outside basically think that they're a virus entering a cell, and so get cells entering each other, which just ends up fusing them together. And they can live for a while. It's a way of spreading the infection. Things like RSV, respiratory syncytial virus, it's because you spot these in the lung. That's why it's named that. Spicules tend to be either spongin or silicon dioxide in demosponges. They tend, there's occasional calcite, I believe, at least in the exoskeleton. There we go. Calcarean spicules are calcium carbonate, I believe, solely. And silicon dioxide is what you get in hexactinellids. Homoscleromorph sponges only have the silicon dioxide so far. And there are old sponges and a couple of modern ones that are classified as stromatoporoids, which we think are probably just a branch of demosponges. We don't have good genetics on these yet that I have seen. So it may be that they look like demosponges but are genetically different. But these are a big part of the fossil record. They were just massive chunks of what looked like solid rock, but it was rock that was filtering water. And yeah, there were sponge-like animal traces inside of it. So pretty cool. Exoskeleton sometimes, but not always. So you'll have a few that have like a calcium carbonate, just sort of wall on the outside. But it's pretty common in Calcarea. And you can see most of the species are demosponges. Demo, it's the same root as democracy. It's like it's the people's sponge, the numerous kind of sponge. What about sponges over time? I hear you asking. This is a fossil class. Yeah, let's get away from these things that still exist. Ew. Yeah. Well, here's some reef builders, and you can see early on, the very first reefs are almost completely, let's say 90% sponge reefs. And then in the lower Paleozoic, it's maybe half sponge and half other stuff. Bryozoa are probably number 2. Corals are there, but they're really not. They're just sort of little single individuals, more than massive joined up corals in most cases. And then you get some big extinctions happening, and the reef builders actually disappear for a short time between the Permian and Triassic. But this is about where they were, and this is about where they bounce back to after a few million-year gap. So it's still about half sponge, and then you see the corals really take off. So corals, as we know them, they're called scleractinian corals. These are a product of the Mesozoic, the time of dinosaurs. So yeah, who cares about T-Rex? Where's my Jurassic Park with the, Mesozoic corals in it? Yeah, that's what I want to see. That's what I want at the cinema. Yeah, none of this Marvel nonsense. There's enough natural marvels out there. Here's the present day. Digression aside. And sponges are a very, very small part of modern reef ecosystems. Yeah, that's just the way it is. It's the way it goes sometimes. You're doing well and then you're not. Speaking of that, look at the Archaeocyatha. Name means ancient cup. And we have some fossils of those in the teaching collection, so you're going to see those. That's what they look like. So these things were doing extremely well. Many different kinds in the early to mid-Cambrian. By the late Cambrian, a bad thing happens right around the middle of the Cambrian, and they almost go extinct. And then at the end of the Cambrian, they do go extinct. Yeah, they are wiped from the fossil record forever. But they were really neat while they lasted. We're not sure exactly on the points where these things diverge, and honestly, this is an older graph. We could probably do a little better now. But here you can see the stromatoporoids, which are these big rocky lump sponges. Calcareous, probably, yeah. Okay, here's an early demosponge. We think this one is kind of Ediacaran, late Precambrian. You see it's got, here's the middle part of it. It's got little pores in there where water could have entered. And there's a big hole at one end, if I remember right, of this one. And so, is that a sponge? It's something. It's probably biological. It's there in a marine environment. It sure could be, but I don't know that I'm that convinced. This is one of the ones that held the record for earliest maybe sponge before that beautiful one that we showed during the earlier part of the lecture. Here are some Precambrian sponges, and these are little tiny sponges. So they're saying this is the sponges, like little holes in the sponge where the coniflagellates were, with some undigested debris, which it might be, yeah. Or it might be debris from the cells themselves, or it might be debris that was introduced later during the fossilization process, during the process of getting buried alive, if that's what happened. Yeah, all kind of iffy, but it was a good paper at the time. Here are the full bodies of these things. You can see it's got some big holes and it's got some little tiny holes. And so theoretically, I guess it could take water in at the sides, for example, and then pump water back out the big hole. Here we go. Could pull in a little bit of water from all these little gaps in the side, filter it in these little chambers, and pump it out in the middle. It's not a lot to go on. It's a vaguely sponge-shaped morphology, and there's no real organization, which is characteristic of a sponge, but also of anything that lacks organization. It's not a definitive characteristic. There would be, potentially, molecular fossils associated with these, but you would have to find a bunch of them, which they haven't yet. They've only got like these couple. And you would have to pulverize them, and you would have to run them through some really cool separators, and then hope that the molecules retained enough of their original structure for you to be able to tell what they are. So it's a bit of a gamble, and it's not what you'd want to do with the first fossils that come out. But eventually, we may be able to answer these questions, which would be to say, did this thing, which is vaguely sponge-shaped, also have a sponge-like biochemistry? Did it make the particular version of cholesterol that some modern sponges make, but nobody else in the animal kingdom or plant kingdom makes? So potentially an answerable question, yeah. Now let's look at some neat fossil sponges, because they're cool. Here's a sponge from the Burgess Shale. This is Choya. And here they are. They got little spikes coming off of them. Look at that. Really gorgeous. We think this may have been a hexactinellid glass sponge. But because it's preserved in the Burgess Shale, all the hard parts are gone, and we only have a mineralized record of where they used to be and their imprint. So you can see a hollow where there used to be a hard part. Here's a neat one. Look at that. Can you tell what kind of sponge that would be? Let's look in here and let's count the little spikes. 1, 2, 3, 4, 5, 6. 6. There's only one group that has six-fold symmetry. The Hexactinellids. 6 rays around the little spine. There you go. So this is a primitive, presumably you would call this a hexactinellid sponge. Although you've always got to be careful with crown groups because they may be far enough down that they're actually the ancestor of modern Calcarea and hexactinellids. I think this one is not. This one's from late enough that those two had already diverged, probably. But yeah. Complex. Here's one, a sponge from the Burgess Shale. And look at this. So we've got traces of where these spicules used to be. And here I see one that's 1, 2, 3, 4, 5, 6. So six-fold symmetry. That says hexactinellid to me. Those were the criteria on which we called this one a hexactinellid. But look in between them. These little ones are just like little X's or little plus signs. Those have four-fold symmetry. And that's more like what you would expect from a demosponge or a calcarian occasionally, although Calcarea would tend to have a lot more 3s than 4s. So this thing may be, and this is down in the middle Cambrian, so it's the right time, but it may have some of the characteristics that we associate with modern hexactinellid and demosponge groups of sponges. So isn't that cool? And so once again, the idea that evolution is like a winnowing. You're paring down all these possible traits. And the early organisms are just kind of doing whatever, you know. They're just expressing all the genes they've got, and if there's any advantage to it, that's great. Later on, we're going to refine by getting rid of a lot of things and just keeping a few of the genes, or fewer of them, making more copies of those. And yeah, we're going to narrow the possibilities of what life can be. Spongy life. Here are some Archaeocyatha. This is what they look like on the outside. Their skeleton, or the epithelium, if you like, it has no spicules in it that we can find at all. It appears to be massive calcium carbonate, and there are these little crystals of calcium carbonate in there. There's an idea that they used to be amorphous calcium carbonate, which is a kind that's maybe a little easier to lay down, but is more prone to dissolving and recrystallizing as polyhedra of calcium carbonate. So it's possible that this is altered. This is diagenetic. That's our fancy term for altered by time and rocks. But yeah, and they have these little things like roots, radules to probably hold them up in the sediment. But yeah, little free-living filter guys. And you can see there's little holes running up and down the side where they can pump water. There's little chambers here, which were probably lined with those little collar cells, which were generating the current to bring water in, because otherwise this thing doesn't work. And you got a little outflow hole on each of those cells. So that's where the water goes out after it's been sort of turbulently filtered in there. And then you've got a central chamber where the main current of outgoing flow is going to be. Here are some other beautiful Archaeocyatha drawings, colors supplied by DeviantArt. Actual shape of these is supplied by the fossils themselves. And so just looking at some of these different types of Archaeocyatha, yeah, and the way they sort of build up little reefs in between other things. Here's a little Archaeocyathal reef, and here's a nice little fossil. It actually looks very similar to the ones that we are going to look at in class. Okay, then we're going to have some specialized ones. We've got a whole talk on these guys coming up. These are called chancellorids, and they're related to this thing called Chancelloria, Chancellloria. And this is Chancelloria, and Chancelloria pentacta has some spicules that have 5-fold symmetry, but it has a lot of them that have 6-fold symmetry and some that have 7-fold symmetry. So it just wasn't too careful about what it was. They look a bit like spicules, but they are hollow, whereas sponge spicules are always solid. So this is a thing with a big hole in the top and presumably lots of little holes on the side, and things like spicules holding it together. Rooted to the ground, you can find it next to some authentic sponge fossils. So is it a sponge? Is it related to sponges? We'll see. We'll see. Stromatoporoids are an awesome group of actual sponges, and there are a few things that, if they were found in the fossil record, would be called stromatoporoids. They've located these on the seafloor. I feel like Jacques Cousteau actually found one of these, if that name means anything to you. Yeah, they're awesome colors, but they're built up of little layers. So they grow by a little layer each time. They'll lay down another layer of rock over themselves, but they'll always maintain these little paths through there where the water is going to be filtered. And there'll be little chambers along the way, and that's where the little collar cells are going to be that are doing some of the filtering. So we didn't know what these were. There was a lot of controversy about what they are, but they have been recently recognized, like in the last maybe 40 years or so, as being actually sponges, definite sponge grade organisms. Yeah. And so they'll have these little mamelons. It's like a little bump that has a hole in it, and sometimes they'll have little ridges running around the hole, and that's going to be where water either is pulled in or where water is pumped out in these. Here is a kind of a weird Devonian stromatoporoid, so the middle of the Paleozoic. And look at this thing. It stands up tall. It's kind of hollow in there, but the organization of this is still kind of solid, and it has been classified as a stromatoporoid. It could pass for essentially a sponge. When it is young, when at the early growth stages, it looks a little different. It grows into more of a standard thing, I think. Yeah. Here is one of the modern stromatoporoid-like creatures from the bottom. Here's another one. And so these are the things that we think are probably equivalent. As of the time I pulled these pictures, there was not any genetic information to indicate whether these are different from other demosponges or Calcarea, calci sponges. Yeah, it's a good question. That may actually be solved now. I just haven't seen the paper yet. Here are some Calcarian fossils, and these come from the sponge gravels of Oxfordshire, which you can buy. And the beautiful thing about those, I guess, is that when it rains, it dissolves part of the calcareous skeleton of the sponges, and then the calcite will spread out and will make more links and essentially cement itself together. It is a self-cementing path gravel, but the thing that self-cements are the fossils of these little sponges. And so honestly, I'd rather they were all in a museum than in somebody's, walkway, treasured walkway. But I get it. Everybody got to make a buck. So I've spent a fair bit of time walking along some of these paths and scratching up little scratch and dent versions of these sponges. Yeah. So there's a beautiful blue sponge on the ocean bottom, just to give you something inspirational. If this wasn't inspirational enough at the top, I think that's enough about sponges sensu stricto, like in the strict sense. We will now talk about chancelorid sponges. That's going to be the next one.